This is the last of three mini lectures that will be addressing these three objectives for learning about the adaptive immune system. This mini lecture will discuss B-cell responses. Here's a brief outline of what we will talk about in this lecture. We will discuss the mechanisms required for B-cell activation and the role of T-cell help in that process, as well as the functions of the germinal center, including isotype switching, affinity maturation, and the formation of long-lived plasma cells and memory B cells. And we'll also discuss the key antibody effector functions mediated by these antibody responses. So here's a picture of a dendritic cell that has taken up antigens and is migrating via lymph to the draining lymph node. And this is an activated dendritic cell that is activated by its inflammatory environment. But within that environment, we also have pathogens and pathogen products that are also drained via the lymph and drain into the lymph node, and some of these antigens make it into the B cell zones. Now, what is the nature of these antigens? Well, B cells can respond to many types of molecules. And so I've shown a carbohydrate chain, a nucleic acid, a globular protein, a couple of globular protein structures, viruses, bacteria, glycolipids, and it's important to note that B cells can really bind to just about anything and make antibody responses to just about anything. However, protein antigens typically induce the best antibody responses, and that's because only protein antigens can mobilize helper T cells. So here's a B cell that's been activated by a virus. When a virus enters into the B cell follicle and binds to the surface of naive B cell, it induces its activation. And this activated B cell can undergo a limited amount of clonal expansion and form short-lived plasma cells. And these short-lived plasma cells almost exclusively produce IgM. And so the very first antibody responses that are induced by an infection are IgM antibody responses and are derived from these short-lived plasma cells. This process does not require T-cell help, but it doesn't induce the formation of germinal centers, and it doesn't induce long-lived antibody responses. For that, you need T-cell help. The other point about these responses is that, like T cells, B cells can receive co-stimulatory signals. But for B cells, these co-stimulatory signals are often inflammatory mediators. So, for example, binding of toll-like receptor ligands to bacterial products like lipopolysaccharides, or binding of complement receptors to complement fragments that are induced by complement activation, can serve co-stimulatory roles in enhancing the ability of B cells to make these short-lived antibody responses. Now, some B cells that bind to protein antigens, rather than making these short-lived plasma cells, internalize these antigens and degrade them, and eventually present them on the surface of MHC class II molecules to T follicular helper cells specific for that same antigen. And these T follicular helper cells do two things to help mediate the activation of B cells. The first is that they express CD40 ligand, and this binds to CD40 on the surface of these B cells. And this co-stimulatory pathway, as for macrophage activation, is also important for B cell activation. The second thing that these T follicular helper cells do is they secrete cytokines. Now, these cytokines differ based on the flavor of the T helper response, but can include things like interleukin-4, interferon gamma, interleukin-21. And these cytokines are important in driving isotype class switching. So T-cell help is mediated when B cells internalize these antigens, present them to T cells on MHC class II, and receive help via the CD40 ligand, CD40 co-stimulatory pathway, and via the production of cytokines. Okay. So when this happens, these B cells undergo massive clonal expansion, and this leads to the formation of the germinal center. Now, there's some really important things that happen in the germinal center, and two of these key functions are induced by an enzyme called activation-induced deaminase, or AID. AID is activated by CD40, and when AID is activated, it goes to the nucleus and mediates isotype class switching of the immunoglobulin constant region and somatic hypermutation of the VDJ region of the immunoglobulin gene. So what does that look like? Well, first, let's talk about isotype class switching. So here we have AID that induces a DNA recombination event that allows expression of other constant regions besides the IgM constant region. So, for example, it can mediate a recombination from expressing the mu constant region to here expressing the gamma-3 constant region. And this would result in an immunoglobulin locus that looks like this. You've got the VDJ, and now you've cut out the mu and the delta constant regions from this locus. And this is a one-way event. This DNA has been cut out. And so you can't go from being an IgG producing B cell back to producing IgM. Once you've lost this IgM constant region, you're never getting it back. And so here we have a B cell that will now produce IgG. And it's important to note there's actually several subclasses of IgG, IgG 1, 2, 3, and 4. We won't discuss really in any detail in this class the different functions of these subclasses, but they all mediate key effector mechanisms. Or this recombination event may go here, and you would instead get expression of IgG 2. And again, you lose all of these intervening constant regions. Or this can happen in a two-step process. For example, you may get a recombination event that leads to the production of Ig alpha, and then a second recombination event that now leads to the production of IgE. And so each of these isotypes is dependent on these DNA recombination events induced by AID. The second thing that AID does within the germinal center is that it mediates random mutagenesis within the VDJ region, within the variable region of the immunoglobulin. And this is called somatic hypermutation. And hypermutation because of the rapid rate of mutation within this region. Most of these mutations will actually disrupt antigen binding affinity, and that's bad for the B cell. But some of these mutations may actually increase the affinity of the B cell receptor for its antigen. And so these B cells undergo a process of selection. So B cells that undergo somatic hypermutations that reduce their affinity undergo apoptosis, whereas B cells that undergo somatic hypermutations that increase their affinity undergo further rounds of proliferation and expansion. And what this does is multiple rounds of affinity maturation lead to the selection of ever higher affinity variants. And eventually you get the emergence of really very high affinity B cell clones. So the antibody responses early during an immune response are generally lower affinity than the antibody responses later during the immune response. One of the key mechanisms that promotes this process called affinity maturation is the retention of antigen within the germinal centers by these cell types, follicular dendritic cells. I talked about these briefly in a previous lecture on lymphocyte development. And what you'll notice is these follicular dendritic cells here, this follicular dendritic cell here is shown retaining these viral proteins. There's a variety of mechanisms by which these cells retain these antigens. But the important thing to note is that it can retain these antigens even long after the pathogen has been cleared. And so it allows for this process of affinity maturation, somatic hypermutation followed by selection of high affinity variants, to occur for weeks to months after the pathogen is gone. And very high affinity IgG often takes months to fully mature. One quick reminder, these follicular dendritic cells are not related to dendritic cells. It's confusing, yes, I know, but these cell types are different. This is not a hematopoietic lineage. And the end result of this whole process of somatic hypermutation and affinity maturation is two things. One is the formation of memory B cells. These memory B cells are mostly class switched, and they're capable of rapid recall responses, so that if you ever get infected with the same pathogen again, you get a rapid boost in antibody titers and successive rounds of affinity maturation. And so if you look at primary versus secondary versus tertiary activation or boosting of B cells, you find that the antibodies increase in numbers, but they also increase in affinity. The second cell type that results from these germinal center reactions are the long-lived plasma cells. And these secrete copious amounts of antibody. They can persist for years. They reside in the bone marrow and are the reason behind why, after an infection is resolved, antibodies for that that infection can persist for long periods of time. So antibody responses become long-lived. Just to sum up, a single exposure to antigens with no T cell help will result in the formation of short-lived plasma cells and short-lived IgM responses. But a single exposure with T cell help can also induce these short-lived IgM responses, but will additionally induce long-lived IgG or IgA or IgE responses. And a second exposure to antigens will activate memory B cells to in turn induce a second round of long-lived plasma cells. And these are going to be mostly IgG producing plasma cells. This second exposure will boost the amount of antibody that's circulating, and it will boost the affinity of that antibody. And you'll generate more memory B cells from that second exposure. So here's a summary of everything that we've talked about. Within the T cell zone, naive T cells get activated by dendritic cells presenting peptides from a pathogen. This leads to the formation of T helper cells, which then migrate to the inflamed tissues, but also leads to the formation of T follicular helper cells. Within the B cell follicle, naive B cells can be exposed to antigens that directly activate them and induce them to form short-lived plasma cells that produce IgM. But these B cells can also internalize these antigens and present them on MHC class II, which allows T follicular helper cells to interact with these B cells and enhance their activation by binding CD40 ligand on T cells to CD40 on B cells and secreting cytokines that induce isotype class switching. So all of this together induces the formation of a germinal center, which is characterized by clonal expansion, affinity maturation due to somatic hypermutation, isotype class switching, the formation of long-lived plasma cells, and the formation of memory B cells. Now, what do these antibodies do? This will be a little bit of review for you, but I want to make sure and talk about this. We've talked a lot about what antibodies do throughout our discussion of the innate and adaptive immune response. One key function of antibodies is they neutralize, and all isotypes of antibodies can neutralize. This can take the form of antibodies that prevent viruses from infecting their target cells, or antibodies at mucosal surfaces that prevent bacteria from colonizing epithelial cells, or maybe even blocking bacterial toxins or other bacterial products. A second key effector function of antibodies is opsonization, and opsonization is mediated by IgG. And this is because many phagocytes express receptors for the constant region of IgG antibodies, and these are called Fc gamma receptors. And this enhances the ability of these phagocytes to take up these antibody-coated pathogens and destroy them within the phagolysosome. A third key function of antibodies is the activation of complement. The two isotypes that activate complement are IgG and IgM. And when complement is activated by antibodies, it leads to the formation of the membrane attack complex and lysis of the target bacteria. It also can opsonize these bacteria. Remember that one component of complement, C3b, can coat bacteria and enhance the ability of phagocytes to recognize via complement receptors those bacteria and destroy them. And lastly, there are complement fragments such as C5a and C3a that induce inflammatory responses and capillary vasodilation. So these are all key functions of antibodies. Third key function of antibodies is the activation of mast cells. We haven't talked about this directly, and we will talk about this more in our type 1 hypersensitivity TBL next week. But there are a variety of pathogens that can, or a variety of antigens that can induce IgE responses. So these include many allergens, many helminth antigens, and certain types of inflammation can induce these IgE responses. And so on a primary exposure to, for example, this pollen, you can induce activation of Th2 cells that produce IgE. And on a secondary exposure, these IgE molecules have bound to the surface of mast cells via binding to these Fc epsilon receptors, these receptors that bind to the constant region of the IgE. When these IgE molecules bind to their antigen, This induces degranulation of this mast cell. These granules are full of histamines and leukotrienes, lots of things that are important for the anti-helminth response, but in our case, it's probably more relevant that these induce the allergic response. And lastly, antibodies mediate antibody-dependent cell-mediated cytotoxicity, or ADCC. And this is mediated largely by IgG molecules. And this happens when natural killer cells express Fc gamma receptors that bind to the constant region of IgG molecules. And let's suppose that these IgGs are specific for a viral glycoprotein. When viruses infect their target cells, they often leave copies of the glycoprotein embedded in the membrane. This allows this natural killer cell to recognize this target cell, and it mediates killing of this target cell by the release of cytolytic granules that contain granzyme B and perforin. And so the mechanisms by which a natural killer cell kills its target cell is the same mechanism by which cytotoxic T lymphocytes kill their target cells. Okay, so that is it for B cell activation and the effector functions of antibodies. Please take a moment to review the key points from this mini lecture as you go through those points summarized here.